mention just a few house rules for the program. Sorry, let me just get this. Okay. Firstly, the event is recorded. In terms of complying with the PAPIA Act, kindly note that we will use the information from the event for publishing purposes. Should you not want your information to be published for the purpose stated above, kindly send your concerns to rajpal@ukzn.ac.za. During the course of the event, all attendees will be muted with their video switched off. If you have any questions, please use the chat box facility and pose your questions, where there will be somebody who will answer them. During the Q&A session, the presenter will give you a chance to ask your questions. Please raise your hand so that the tech person can allow you to unmute you and pose your questions to the presenter. If by any chance attendees interfere with the presentations, we will note your details and remove you from the event. Finally, the recordings will be emailed to all attendees present, and we will also post it on the CAES website. I would now like to hand over to Dr. Nontuthuko Maningi, who is from the School of Life Sciences. Thank you. You're fine, Nontuthuko. You can go ahead.
Fine. Yes, good day. It's not morning. Good day, everyone. To our postgraduate students and our colleagues. I would like to welcome the team from Inqaba Biotec. And I would also like to firstly thank Leena and your team for organizing these industry talks for our students. And I would like to assure our students that for attending this talk, they will not regret it. I know a lot of students are working hard trying to obtain their postgraduate degrees. However, in the back of their minds, they've got numerous questions. And some of them are questions like, then what? Then what after obtaining this degree? Where to after obtaining this degree? So these industry talks will help our students to know, to answer those questions that they always have in the back of their mind. They will also answer those questions on how to find employment, where to look for employment. I would like to assure our students or our postgraduate students that they are very important and they are highly regarded in the industry because they are young researchers and they've got a responsibility to transform science skills. They have a responsibility to transform technology skills in the industry and engineering skills. So they are very important and everyone is looking out for them. And then I would like to welcome our first speaker for the day, which is Mr. Moila. Mr. Moila is okay, let me open. Mr. Moila will, okay. She's going to tell us about the company profile, and she's also going to tell us how various academic fields such as IT, health science, biochemistry, microbiology, chemistry, etc. converge at Inqaba Biotec. Over to you, Mr. Moila.
Thank you. I'm not sure if you can just allow my video.
Sneha, just allow, make Acclaim co-host. I think he dropped and came back.
Yes, he is.
Okay.
All right, great. All right, thank you. It's really a great pleasure to be here to share some information with all the students of UKZN. And as a company, we really do more than just being in business. it is really kind of our responsibility to try and transfer knowledge and also to train as much as we can the students, as we usually do. We take a lot of students for that work for us. It's a very young group that do all this work. So we believe this would be very much beneficial to you as well. So I'm just going to share my slides here. Right. So basically, I'm just going to go through our company portfolio, as mentioned already, just to guys kind of get a good idea as to what kind of services we do and how do we do all those services. I'm sure a lot of you, some of you have worked with us before, either at the research level or just undergrad level. So basically, our company this year, we're turning 20 years. So we started in 2002 in August. So we are 20 years this year. It's been a really great journey for us. But what started really the business when we started was the oligo production as well as Sanger sequencing in 2002. And in 2007, we became the first company in Africa to offer the next generation sequencing services. And then in 2010, we opened our East Africa subsidiary, which is based, the main office is in Kenya, Nairobi. And that was followed soon after that in 2015 by the West Africa subsidiary, which is the main office is in Nigeria, Ibadan. And again, in 2018, Ghana office was opened, 2019, the Tanzania office. And then in 2020, we really kind of partnered with the company known as PacBio, which is a US company, which does sequencing, the long read sequencing instruments. And we started to offer such services in 2010. So we have seen from 2020 until now, the shift is really going towards the long read. And this year we registered our offices in Uganda and Senegal as well. So basically, our footprint is increasing in sub-Saharan Africa, but our main headquarters are in Pretoria, just not far away from UP actually, the main campus. You can see here, so every flag here is represents our presence. And then this is really some of our employees. I think when we started in 2002, there were like 5 employees. And now we are over 150 across the sub-Saharan Africa. So we've really grown since then. So like as I mentioned, our goal really is to provide services to researchers across the sub-Saharan area. So we also, apart from just offering, so basically we are, if I can just put it away, we are a two-hand company. So we, on one hand, we offer the services, and then on the other hand, we distribute the leading brands in life science and biotechnology. So we have a huge portfolio of brands that we distribute, but I've just put a couple here for the main ones that we distribute, like for PacBio, New England Biolabs for your PCR reagents, and Appendorf. I'm sure everybody knows Appendorf for instruments, pipettes, and tips. Zymo Research for your DNA extraction and RNA extraction and purification of amplicons. The CG for our diagnostics, such as testing for COVID, and then TREF for plastics as well. Could be your PCR tubes, 1.5 mil a piece, stuff like that. So like, as I mentioned, when we started the company, when it was started in 2002, we're mainly offering oligos. So we have since upscaled the production level by acquiring the system that is able to produce up to 1,000 oligos per day. And then due to the increase in demand of the primers, which we saw really when the pandemic started, we are very, very much reliable in producing those oligos to supply to researchers. So we are really producing them, and our turnaround time was really quick. So that kind of helped us. And then now we're at a point where we got a new instrument as well, just to increase the throughput. And then this specifically will be used to synthesize the probes. And then we have another department, the Sanger sequencing department. So basically, anyone that wants to do bacterial, fungal, or animal species identification from pure cultures or tissues, you can easily do that with Sanger sequencing. Or if you want to sequence any a known or a single amplicon generated from the pure cultures I've mentioned, you can easily use the Sanger sequencing platform. And then we do also have the animal genetics department. This is which is known as Genomics now. So we do parentage, a whole lot of testing, and then we have recently actually when we came up with the name, so we kind of combined with another company that does animal genetics, so that we can offer holistic animal genetics test. We can do custom tests as well and develop new tests based on the customer needs. And then we also have a department that does SNP genotyping, targeted SNP genotyping through the use of the mass array system. This is a very simple, reproducible PCR-based chemistry. So how it works basically is, if number one, we need a very good genomic DNA or specimen, and secondly, since it's a targeted SNP genotyping, that means one has to know their SNP ID, or at least the sequences indicating where the SNPs are. For instance, if you don't have the RSIDs or SNP IDs, you can give us a sequence as indicated in this way. So so you can give us the sequence as indicated here, indicating where the SNP is, but there must be at least 100 base pair flanking both ends of the SNPs to allow the design of the assay. So how it works is basically you have a genomic DNA. We design the primers targeting that SNP. So basically we design 3 primers. The first primer pair is basically just to enrich, just kind of enrich the target. We do PCRs over limited number of cycles. And then we do purification. And then the other primer, which is very important in this case, is called the extension primer, which basically just sits adjacent to the SNP, so that we only have a single base extension. And that single base basically is what identifies the SNP, what SNP is. And then we take that reaction and put it into the instrument, so the multitoph, and then we're able to pick up the SNP. We can also do targeted methylation on the same system. Like I said, you have to know the regions of your interest. Then you pretty much follow the same procedure. We design primers to enrich the target before we do the SNP calling. And then also, she's pretty much plays a huge role in our portfolio, the next generation sequencing platforms. So we have 2 platforms: the Illumina platform, which is the short read technology, and the PacBio SQL 3E system, which is the long read technology. So basically, when you do Illumina sequencing, obviously you start with the library prep, and at the end of the day, what you want to end up with when you create your library is you want to have a structure like this, where you insert DNA, which can be roughly between anything up to 150 base pairs, which is flanked by a sequencing an adapter, which includes the barcode, the sequencing primer binding site, your barcode, as well as the P7 adapters, which are complements to the sequencing flow cell. So during sequencing, obviously the flow cell will have complements of the complement oligos of the P5 and P7 adapters, and then this is how the molecule kind of anneal to the flow cell, the anneal upright. So you'll have your P7 or your flow cell oligo kind of annealing to the P7, and then you have an upright molecule. Then in a second cycle, then you anneal. There will be a sequencing primer annealing, and once the sequencing primer annealing, and then you have all these fluorescently labeled DNTPs that have kind of reversible terminator molecule. And then as the polymerase, we basically call it the sequencing by synthesis, because we detect, we can get the base calls in real time as the polymerase extends the chain. So you have a nucleotide that will bind, and because it has a reversible terminator, this prevents a second nucleotide to bind right immediately. So you have a polymerase that binds the nucleotide, a particular nucleotide. A light will be emitted, that light will be captured. And once it's captured and that reversible terminator is removed, and then this allows the second base to be ligated, so on and so on, until you have a growing chain. And this happens in two directions. Once you have achieved, like for instance, as I mentioned, 150 base pair in the forward reaction, the molecule will flip over so that you can have the reverse strands sequenced as well. Now, how do you differentiate this with the long read technology? It's basically the main thing here is the insert sizes, the short reads versus long reads. That means with the long read, you can sequence anything from 250 base pair to 20 kb long fragments. And then we call those reads the high-fi reads, high fidelity reads on PacBio. These are basically produced through the circular consensus sequencing mode on the system. And then they are pretty much as accurate as the short read fragments. And then you can use this for a wide range of application as well. So how do you get this hi-fi reads? So you have your genomic material. You share the genomic material to roughly 15 to 20 kb long fragments. As with any NGS library prep, you have to ligate adapters. So you ligate adapters at the end of the fragments. These are the same adapters. And then once you've ligated your adapters, you move to the next step, which now anneal the sequencing primer. So you anneal the sequencing primer there, and immediately after that, you bind your polymerase. This is done outside before you even go to the system itself. So what you take to your system, to the system, is you are loading a bound complex, which is already primer annealed and has a DNA polymerase bound. Now, how does the sequencing take place? So these molecules during the sequencing will kind of open up in this fashion, become more like a circular structure, and the polymerase will go over that molecule multiple times, generating all these subreads, forward and reverse, forward and reverse, forward and reverse, including the sequencing of adapters, as you can see in the ends here. Now, what happens post the sequencing is the hi-fi processing. So because the sequencing here is sequencing multiple fragments multiple times, and we want to produce a very high quality long read data, all this produced subreads, we then kind of collapse them into a single consensus read, generating what we call a hi-fi read. As you can see here, you have kind of red dots there and there, and this is the nature of polymerase. You get random errors, but for the mere fact that these errors are random, when you combine all these reads to generate this hi-fi reads, you literally flush out all the random errors, and then you end up with a very, very high quality long reads. So basically, that is the difference between the two systems. The application that you can run on both systems are pretty much the same, depending on, of course, the goal of the research. Yeah, in a nutshell, that's all our services that we can offer. And yeah, I'll be happy to take any other question after that.
Thank you, Mr. Moila, for that great talk. I forgot to mention at the beginning that we're going to open a Q&A session after the presentations. But for so long, anyone can put their questions under the chat box.
Right.
Okay, I'm sure our students have burning questions on how they can purchase consumables, equipments, and how they can also get services on NGS.
Right.
Thank you so much, Mr. Moila. And then now I'm going to move to the next speaker. The next speaker is Dr. Arndt. Dr. Arndt is an NGS manager at Inqaba Biotec. And Dr. Arndt is going to tell our postgrads how they can get jobs at Inqaba Biotec. Dr. Arndt, over to you.
Sorry, I'm struggling to share my screen. I'm trying. Let me see.
Okay.
I did say, can you see my screen?
Yes, yes, I can see.
Okay. Okay, good.
Thank you.
Welcome, everyone. So my name is Silke Arndt, and I'm the next generation sequencing service manager for Inqaba Biotec. And I want to walk you through how we recruit people in Inqaba. And the good news is we have a lot of young people working in our company. So currently in Pretoria, we are about 80 employees. And as you've heard, we started out with 5. So a lot of recruiting has happened over the years. And we also recruit in a fairly diverse portfolio. The reason for that being, as you've heard from Acclaim, we basically have a few streams within the company. So we do sell life science products, and obviously those products have to be moved. And so for that to happen, we have a very strong logistics department. And for the logistics department, really what we are looking for is people that have at least a matric, so they are not required, there are no degrees required. And if they have work experience, that's even better. And we train them in our logistics portfolio in-house. So then the second branch of the company is, is sales. So as we sell these products, and that is not restricted only to life science products for the laboratory, as you've seen, we also sell instrumentation. We are in the sales force really looking for people that do have a life science background or a degree. So in most cases, if you want to sell molecular products, for instance, it's absolutely vital that you have a basic understanding, and we recruit people from the life sciences, biochemistry, microbiology, genetics, that would fit our portfolio. We always train people, so they are not expected to arrive here with all the knowledge, but they obviously have to have some background. And then for our platforms, we have a certain engineering support department, and they go out and support all the platforms, repair or calibrate, or that we have delivered around the country. And this workforce would come from an engineering background. So, and they also get trained in-house. Then we have the second branch, which is services in the company. And services, all our services are laboratory-based. And there again, we look for people with a life science background. And in most cases, almost in all cases, we train the people, but they do have to have, number one, shown an interest and be engaged with the field, and also show initiative to learn in the field. So we have 4 laboratories actually. We have the Sanger sequencing laboratory, where both things are done. You can do laboratory work, or you can be involved in data analysis once you know the lab processes and have been trained in addition to what you've known before. Then the Genomics department, again, it's lab and there's lab and data analysis opportunities. Then we do a gene SNP typing. Acclaim spoke about that. Again, that is primarily lab-based. And then next generation sequencing, which for young graduates, it would usually be starting off in the lab, unless you know, they have done some additional qualification, and that would, for instance, be a bioinformatics degree, where they learned how to analyze data. But all of this is targeted at life science students, and we regularly take in young graduates to train them in our field. And then we also have oligonucleotide production that Acclaim has mentioned. And there, this is more production based. So it's you've seen the manufacturing device, and it's a lot of quality control and, you know, driving of the production, and that would be suitable for a chemistry or biochemistry background. And last but not least, we obviously, the company needs to be administered financially and human resource wise. And in that area, we would look for young graduates with a financial degree or some work experience that can be built on. Okay, so those are basically the areas of work within the company. You can see it's quite diverse. The absolute majority of our employees come in and start off with an internship at Inqaba. They obviously need to know where they would like to be and what their interest is, and you know, possibly so the internship route is usually we receive your inquiry. If there is an internship opening, then with a full CV and a letter of motivation and possibly your degree certificates. And if something opens up, we basically look in our application pool and we contact people that seem suitable for interviews. And then it would start out as a 3, in some cases, longer internship. Also, it gives both sides an opportunity to find out if that is actually what they were expecting or wanting to do for the next few years, or however long it might be. Many of our interns have most of our interns actually have grown to be, or many, I should say, have grown to be full employees of the company, and they are here for years and grow within the company. So we do have, in some cases, positions where we don't have an internship application in the pool, and then we would advertise it in the normal media following national guidelines. So, but I don't think that would be something where young graduates would fit into. And that is how the employment or the recruitment process at Inqaba works. And with that, we will take questions if, yeah. Thank you so much.
Thank you so much, Dr. Arndt. I'm sure now our postgrads know where to check to find vacancies at Inqaba Biotec. Do you also perhaps put your vacancies on your website, or you only put them on the national media, newspapers?
To my knowledge, we haven't advertised on our website, so we do not have a career portal. As I say, we continuously get applications of interested young people who want to join post-university, the industry. And, you know, most often the motivation is to gain practical experience. And so we usually dip into that intern application pool because it shows initiative, and you know, it shows that people have given it thought where they would like to work. So no career portal on our website.
Yeah, just to add, yeah, for those that are kind of not internship kind of looking, for instance, bioinformatics, those we do put on our social media accounts. So if you can follow us on LinkedIn, we do put some advertisement there as well for higher qualifications.
Okay, Mr. Moila, our chat box is flooded with questions, but then I'll start with those that would like to raise their hands and directly ask their questions. Please raise your hand if you want to ask a question. Yes, Snenhlanhla. Snenhlanhla, are you there?
Yes.
Oh, yes. Please ask your question.
Nanto, I think the first.
It's Minenhle.
Yeah. It's Minenhle.
Minenhle, please unmute yourself and ask a question. Okay, I'll move to Olaba Midel.
Okay. Hello, good morning or good afternoon. I don't know if you can hear me. Okay.
Yeah.
My query or question is directed to both Mr. Acclaim and Dr. Arndt. I recently completed my PhD in electronic engineering at UKZN, and I'd very much appreciate the opportunity to learn or be trained and to work in the engineering support department at Inqaba. Is there any chance, are there any chances for me? Do I send my CV to the address sent, or I should just wait until it's advertised, you know, in your portal? I don't know. That's the question I have.
I have to say, a PhD in engineering, you know, it's a very high qualification for technical support. But I would suggest you send your inquiry to info@Inqaba. We will direct it. What does happen is, whenever there's an opening in any of our departments, we basically go back to our pool. So I'm not the HR manager, as you've seen, so I cannot answer the question if there's anything right now. But I would suggest go the route of sending a CV with the supporting documents and the motivation, and it will be forwarded to the relevant department, and they will say if there is a need at the moment or not.
Yeah, I mean, just to add, I think our engineering department is the latest department to be established in our company portfolio. So we kind of slowly growing. I think, yeah, you should give it a shot. We support our area is quite, the area we support is quite large. So yeah, I would suggest that you do indeed forward your CVs there.
Okay, thank you for that question, Ola. Okay, there's Minetle. Minetle is back. Minetle, please ask your question.
Maybe unmute.
Unmute yourself. Okay.
She is unmuted, Nonto.
Okay, maybe she's got a network connection problem there. Let me move to the chat box. There's someone who has asked if the company is looking to expand to other countries in the future, and that question was asked by Jesri Pillay.
Absolutely. So we are expanding all the time, and I think from what you saw with the company overview that Acclaim presented, we started out in South Africa with 5 people, and we now have a substantial sub-Saharan African footprint. In some countries, it is primarily sales, but in Nigeria, for instance, we also have a laboratory. So the company is definitely growing and always looking for new opportunities in additional countries.
Okay, and then Simpiwe is asking if she's asking when do you guys post your vacancies?
As I said, we do not have a career portal that is maintained. If there is a vacancy for, as Acclaim mentioned, for instance, bioinformatics personnel is not very well presented in our intern application pool. And if we need to expand, for instance, bioinformatics, we will advertise it as mentioned on LinkedIn or in the newspaper. So we follow national guidelines there, but that's not that frequent that we look for highly qualified people to start with, and we do not have a career portal on our website.
Okay, and then I've got a student here who is who's asking if the company only recruits postgraduate students. Or
Absolutely not.
also undergraduates.
No, well, usually, so that's why I started in logistics, for instance. So there are definitely, positions in the company that don't require a degree, but the majority of laboratory-based work is done by graduate students, and sometimes in some cases technicons. So we do have also graduates from technicons. So it doesn't have to be university, but it really depends on where you want to join, because bottom line is we don't want to put a person into a portfolio which they then do not cope with, because there is a lack of basic understanding of the fundamentals. Let me put it that way. So I hope that answers the question.
Okay. And then Minenhle has posed her question under the chat box. She's asking if under engineering support, do you consider chemical engineering? And if yes, what are the typical roles of a chemical engineer?
So I spoke to my service department before, while I was preparing this talk, because quite honestly, I don't recruit in that area, so I don't know. But I was, so apparently, as long as it's an engineering degree, it would be suitable for our service department, to support instrumentation in the field. And that is, that goes, you see, we support sub-Saharan Africa. So apparently chemical engineering, mechanical engineering, whatever engineering is suitable. Question is, if a chemical engineer, we obviously don't do chemical engineering in Inqaba, as you might guess. And so it would mean training in a different or in an engineering environment, but not necessarily chemical engineering.
I think also just to add one thing, I think this is probably the only company that does it. So we pretty much believe in, as long as you have the will to learn, we pretty much believe in that. Yeah, at some point you're gonna get to know what you're doing, and you're gonna do it good. So one has to have the heart to learn, because we do training. Some of the guys that work for us, like Silke mentioned, I started in the lab myself as an intern, and look where I am now. So just so it's a matter of the willingness to learn and kind of adapt as well, because we're not just straight and focused like mechanical engineering. You have to go and do mechanical engineering. You can kind of interchange between disciplines within the same field. So for us, if the person's willing to learn and adapt, we can teach that person different things. We have people that did logistics, but they're very good in marketing now, within our company. So if you want to learn, you'll definitely pick up.
Okay, and then the next question is for you, Mr. Moila, from Dineo. She's asking, why do we have Senga and next-gen sequencing labs separated into different departments?
Those are two different sequencing technologies. Samples are prepared differently, and one thing you don't want, especially with next generation sequencing, is contamination, cross-contamination. So we separate two workflows, one to avoid cross-contamination, because there's a lot of movement when preparing the sample. And also, like I said, there's just two different departments. So I don't think, even though they are sequencing, they're two different technologies, and one has to treat both departments separately, because number one, the workflows are different. And with Senga, you don't do RNA. With next generation sequencing, you have to do, at some point we do RNA sequencing, and that has to be separated from any other implicants to avoid what I've just mentioned, cross-contamination. So it's two different disciplines.
Okay, and then a question from Sianda. She's asking if you take people who studied physics.
The question, the answer, we probably do, and then try and find out what that person is good at, other than physics, or what we can harness within the company of that skill set. So would be an interesting letter of motivation.
Exactly.
And then the next question is from Celestine. She's asking if there's any internships open for next year.
Probably, Yes, I didn't interact with the person now, but we are always looking on the lookout for interns.
Yeah.
So we don't say we give 2 per year or 3 per year. As we grow, we take in interns, we train them, and they start participating in production or laboratory service. And you also don't forget, there's quite a training process. It does take a few months for somebody to, in the beginning, it is usually a one-way information flow with participation from the intern, obviously. But it does take time to train someone up for them to become, a valued member and be able to work independently. So the answer is yes. Always try.
Okay. Then another question from IT is asking if there's any opportunities for postgrads with data science skills.
Sounds like definitely something interesting. So as you remember the slide that I showed about bioinformatics, so data science, I understand goes, is that. And so the biological background obviously, would need to be there, but yeah, the answer would be yes.
Okay. And then I've got another student here who's asking if you guys offer vacation work for students, I think undergrad students.
We do. As I said, there were, it doesn't, we should have had a closer look at that big picture from the company, because we have a lot of young people, and we frequently are at events to engage school children would get them interested in the field. And we do accept learners in for, for a stint, I want to say. It varies, if they, depending on what they want to do, but the answer is yes, we do.
Okay. The next question is from Ashik. Ashik started with a compliment. He or she wants to thank you guys for the great presentation. And the question, let me see, what emphasis do you place on social media and community outreach?
I leave it to Acclaim.
Right. Yeah, I mean, if you check our LinkedIn, Inqaba Biotec LinkedIn profile, you will see the activities there. And also, I mean, we even go and reach non-scientific communities where we also participate. Like we have community in vendor that just do what do you call it, clay pots and stuff like that, and we sponsor them. We kind of promote them as well on our social media through Twitter as well, and LinkedIn mostly. So like, yeah, we're pretty much active on our social media. I think, and even on our Twitter, if you go to our let me say YouTube account as well, see a lot of activities there as well. In some areas, other countries, we even have people just specifically assigned to the social media management. So we're pretty much active on that front.
Okay. The next question is a technical question, Mr. Moila from Kanyezi.
Yeah.
She's saying RNA is very sensitive. Now, how do you ensure there's no contamination in the NGS lab?
You just make sure you work where there's no DNA. You just separate the workstations.
Yeah, and there's also RNAs. There's RNA zipper, and so there are things in place to prevent.
So we have a room where only extraction and RNA extraction happens, a fume hood where you make sure that you work in there to avoid contamination. Always clean the surfaces. If in case you're sharing the workstation, there's what RNAs later, something like that, that kind of get rid of all the RNA contamination. So yeah, it's very sensitive, that is true, but you have to make sure that your workstation is separate from amplicons or genomic material.
Okay. Another question is from Nosihe Sbia. She's saying she's more focused on plant biotechnology and mostly on microscopic level. Is there a place for this at Inqaba?
I think, yeah, we are pretty much a biotechnology company. Yeah, that's what it does. I mean, biotechnology is very wide, as you know. So I think, yeah, such person, yeah, there's always an opportunity. Like as Silke mentioned, biotechnology, microbiology, biology, life science, biochemistry, those are the type of people that work for us. Genetics as well.
Okay. There's lots of questions. There's another one from Dineo. She's saying, when looking at the pool of applicants for internships, do you opt for candidates with experience in the field, or does undergrad degree suffice to be considered?
It very much depends on what we are wanting to fill. So undergrads do get considered, but and work experience is in most cases not what we are looking for. We do realize that these people are fresh out of university, and usually, as I said, it's, you know, they have the willingness to gain experience, and they are very aware that this is not, that they don't come with, you know, they know it all. So, as I say, undergrad, yes, we do consider them, depends which portfolio, you know, which part of the company. And I think our salespeople, for sales, for instance, if you are willing to learn bottom line, so if you are, for instance, you know, like somebody who likes to interact with people and who's, you know, good at learning, then that might be a career. So,
yeah, I mean, most of our, I mean, the products that we sell are the product that we use in the laboratory. And most we've seen that it's more beneficial, especially when it comes to sales, the department. So most of our sales staff, most of them have worked in the laboratory, understood the protocol, understood the products. And then that makes it easy for them to kind of move into the sales department. There's no, like, particularly somebody who came with the sales and marketing degree and went straight into sales. So that's how kind of flexible we are. You can start in the lab, and then you can express your interest in sales. You'll be selling pretty much the same products that you've been working with in the lab. So that should make it easy. So, yeah.
Okay, and then last question from me, Mr. Moila. Do you offer trainings, NGS trainings or seminars to students, to a group of students?
Yeah, we do.
That
Yeah, we do on a yearly basis, except the last 2 years with COVID. So we have what we call the NGS Advanced course. It's a 5-day course, where we kind of explain on day one, basically explain the differences, the different technologies in the market and how they're different. And then from there on, basically the next 2-3 days it's hands-on experience. They go in the lab, they do the work, give them reagents, they do the work, of course, supervised. And then the last 2 days, it's just pure data analysis, bioinformatics, because those two really go hand in hand. But what we've seen recently is more, less of kind of hands-on training, but more of data analysis. But yeah, holistically, we do have the NGS courses. I think there's one coming up somewhere in November, if our plans go accordingly. And then, yeah, we do.
Okay, thank you so much to both our speakers. Great presentation. I'm sure the students learned a lot from today. And
Yeah.
I'm sure one of our students will be an Inqaba Biotech employee one day. Not one, but most
Yeah.
of our students.
Right,
Yes.
right. Okay.
Yes.
Yeah,
Okay.
thanks
Thank
for your
much.
time.
And thank you, students, for attending this talk. It was well attended. I'm so proud of you guys. Now, I would like to remind you guys of our next talk. Our next industry talk is on the 13th of September, and it will be offered by Sasol company. Thank you so much. Over to you, Leena.
Thank you, Nontu. Thank you, everyone. And we'll see everyone, our students, at the next industry talk. So thank you so much.
Thank you, bye.
Thank you. Bye.